using the device as well. So encouraging that it seems to be working. Our primary outcome is the Wolf Motor Function Test, which again is a time test. And here I'm showing the change from baseline at 2, 6, and 10 weeks. So down is better. The dashed line is the MCID, which stands for minimum clinically important difference. So that's kind of the standard. If you meet that standard, that's clinically significant. And although it's early, the colored dots here are all the experimental groups. The red is the average of all 3 groups. And the green is the 3D group, which seems to be doing the best at this point. And then the black is sham. So even at 6 weeks, all the groups seem to be so far doing better than MCID, although not quite statistically significant. And encouragingly, at 10 weeks, which is 4 weeks after the training stops, they continue to improve. And that's already significant overall for experimental groups, significantly better than sham, so it doesn't seem to be learn non-use. It seems to be specific to reducing coactivation. Also importantly, all but 3 subjects were severely impaired, meaning Fugl-Myers of less than 25 initially, for those who know. And even these severely impaired patients in here, the experimental group's doing better than sham, and also improving and better than the MCID. So encouraging so far that it improves function. Just got another video here. So we're also, in addition to these clinical measures, we're measuring kinematics of the arm using both inertial measurement units and a VR system called the HTC Vive with trackers here. And we track them while they're doing different reaching movements, both in front, to the side, up, and sweeping out as big an area as possible. As you'll see in this video, this is one of our subjects at her baseline. And she's trying to match what the tech is doing there on the right. And then this is the follow-up video showing that she can sweep out a much bigger area. And this is actually a year later. We just happen to have a nice video of her a year later that shows that the improvements were retained. So this is the ensemble metrics of the kinematics. So and you can see again, not yet statistically significant, but there's a strong trend to having improvement. So here I'm just showing the change at 6 weeks from the baseline. And again, all the colored dots, except for the reach group, are doing better than the sham. Especially the 3D group seems to be doing much better. And in the sweep task, this is an example of that sweep and how we measure the area. Again, all of them seem to be improving over time. So encouraging, and a lot of, and that's important because on these functional tests, sometimes the patients can, again, compensate, right, and cheat, whereas this is measuring kinematics from the endpoint. Sorry, I should have mentioned, we're measuring the endpoint kinematics of the wrist relative to the shoulder here. So if they're leaning over or whatever, that's factored out in our calculations. So this is all actual reach distance. So we've got a second project that's trying to enhance our MINT conditioning, with something called targeted memory reactivation. And this is more recently started, but I'll describe it briefly. This is a collaboration with Ken Paller in psychology at Northwestern, who pioneered this idea that if you pair a memory with a sound or a smell and play back those cues during slow wave sleep or deep sleep, that you can actually enhance the memory. And so, for example, this has been shown in a lot of different memory paradigms. This is a visual memory paradigm where you have to remember the location of the cat and the teapot here. And each one of those is associated with a sound. You play back half those sounds during slow wave sleep and then retest them after they wake up. And what you can see here is error is shown here, so higher is worse. And the cued sounds, they do much better than the uncued sounds. And so this, most of this was tested in declarative memory, and it was thought that this was hippocampal-based phenomenon related to sleep spindles, which are highly related to the hippocampus. We published a study with them last year with the Paller Lab looking at combining TMR with a MyoCI paradigm in healthy undergrads. So here they're doing a variant where they're controlling the cursor to 8 different directions with 4 different muscles mapped in these directions, in these cardinal axes. So they had to combine their arm muscles to reach these different targets. And then we played back half the sounds during sleep. And as you can see here, again, this is time to target normalized path length, so down is better. And the uncued in pink here, the cued in pink improve from sleep. The uncued directions got worse. Same with path length. So it seems to work in motor learning, which suggests that not only could it enhance rehab, but it's not just involving the hippocampus. So we're interested in what structures are involved in TMR exactly. So now we're doing this follow-up study with comparing our wearable mint conditioning at home with home-based TMR, which in itself is also novel because TMR has mostly been done in the lab for a night or most 2 nights. And to see whether over 6 weeks it has any cumulative effects is also a basic science interest of ours. So we're going to be looking at whether that can enhance rehab in our stroke survivors and looking at effects of stroke location and a bunch of other interesting things. So that's ongoing. So switching gears a little bit to BMIs now, we talk about using them to replace and rehabilitate function and a little bit about using BMIs to understand neurophysiology better. So Amy tells me you're all familiar with BMI, so I'll skip this slide, but this is some standard state-of-the-art, or as of a few years ago, state-of-the-art BMIs. And basically the idea is you take signals from the brain, control some external effector. One question is, where do you get these signals from? Most of what you see on TV is using intracortical signals, but that's using spikes from individual neurons. Those are, at the moment, unstable. Pending what happens with Neuralink, we'll see. But for the moment, with Utah arrays, you can record those for 5 years or so, and then they fade out. So a lot of groups have been interested in what can we do with other types of signals. Obviously, EEG is a very popular one, but we've been interested, as Amy has, in electrocorticography and epidural signals. And ultimately, there's probably, depending on what kind of output you want to control, there may be a role for all these types of signals. So just to do a quick survey of some of our older work, we're interested in intracortical local field potentials, which are some potentials from tens of thousands of neurons, so likely more stable. And in fact, we showed that we could use LFPs to control a BMI in the same way that spikes were used to control a cursor, for example, here by building a so-called biomimetic decoder of arm reaches from a monkey on day 0, and then using that same decoder for over a year without any loss in performance. And this was done by Robert Flint, mostly, who was a postdoc and now research faculty in my lab. And we did a follow-up study showing that not only was the performance stable, the signals themselves were highly stable. These are just 2 different measures of stability, which I don't have time to get into, but the bottom line is in hand control and in brain control, the LFPs outperformed, were much more stable than the spikes, and stable for a year. So even without doing any recalibration or doing any kind of subspace rotation, which is kind of the popular thing at the moment, you can just record LFPs and get pretty stable signals. So that does beg the question to some extent, what are LFPs? And specifically, high gamma activation is, or broadband activity, as tends to be said out in Washington. What is that signal is not entirely clear. The field as a whole assumes that this signal from 70 hertz on up is a proxy for ensemble spiking activity, and that's kind of based on these 2 papers by Superteam Ray et al., which looked at, on the same electrode, high gamma activity. As you can see in this spectrogram here, it shows time, frequency, and the increase in power with, in this case, a sensory cue. And this black line here is the spiking rate, and you see that there's pretty highly correlated. And they call this very highly correlated across neurons, although if you look at their histograms, the highly correlated goes up to 0.5 R, which most people wouldn't call highly correlated. So the story, I think, is not as clear as advertised. And we actually did some experiments too long ago now, which have yet to be published, but where we took our orthogonal mapping paradigm and applied it to high gamma and spike. So somewhat similar to what Eb Fetz did, 60 years ago at this point, with one-dimensional biofeedback and what Chet has done with some neurofeedback, but looking at high gamma mapped in one direction, spiking activity mapped in orthogonal directions from the same electrode, so a single electrode here. And then with the hypothesis that if they can separate these things in the same way as we did with the myoelectric control, then this is the closest spike, so it's not just an average of the spiking activity. So, and indeed, we did see that monkeys were able to do this task. And so you can see here, it's not perfectly straight lines, but he's easily able to separate them. And so, and in fact, if you look at the kind of the mean lines over time, they do get pretty straight. And if you look at the correlations, importantly, during the first dot here, this is over a few different monkeys at different channels in each monkey. The first bar here is hand control, and the second bar is during this ONF brain control. And you can see that the correlation decreased over time in all of these. And so they can separate these two signals, and we're trying to figure out at this point what's going on in the network when this is happening. And that's part of the reason it hasn't been published yet. So, so back to what we've been doing more recently with BMIs. Mostly we've been focusing on ECOG or epidural signals. These are attractive for a number of reasons. One is you can do translational work in humans without adding extra risk, because people with epilepsy or tumors often need to have either awake intraoperative situations where we can put a grid down, or they need to have grids put in. And so, and they're more stable and probably better longevity than spikes. And we can actually get a decent amount of information out of them. Plus, they cover a wide cortical network. And that's important, both if you think about Penfield's motor homunculus, so to speak, how big of an area the hand and the face cover in terms of cortical real estate. And this was corroborated by work from Eddie Chang's lab showing, again, speech-related information is over 4+ centimeters of cortical real estate. And if you think about what a Utah array covers, it's this little dot here of 4 millimeters squared. So it's much easier to cover that with ECOG. So we showed, so Robert showed again that we can decode during a grasping task the principal components of finger kinematics almost equally well with epidural signals here in blue as we do with subdural. Subdural did better in general, especially at higher, more complicated functions than just open and close. But both of them did pretty well with, again, not optimally placed electrodes for what we were trying to do, just clinically based placement. We also looked at force and showed that we can decode isometric force with pinch and even discriminate between different fingers with these sort of medium density 4 millimeter spaced electrodes. And so Robert was interested in the difference in encoding between these different states, so kinematics versus kinetics. So he designed a task based loosely on a task that Francisco Valera Cuevas did about 10 years ago, looking at combined movement and isometric phase in the same finger, recording kinematics and recording ECOG at the same time, and then looked at the ensemble activity, ensemble neural activity during this task in the different states. And so what this is showing is, this is a little complicated to explain, but here, these are the low frequency features, so mu and beta bands, so that's everything from 8 to about 30 hertz. And this is the high gamma band information looking in neural space. And this is looking at the neural vector angle in this. So for example, if you have a 64 channel grid, it's a 64 dimensional space. And looking at the angle at any one point in time compared to, of that vector compared to the angle at the peak force in black here. And so the absolute number doesn't really mean that much. But what's important is just how does it change with these different states. And we just wanted to see how it changed. And what we saw is that the low frequency features here they changed right before movement onset here in cyan, but they didn't really change much after that with the rest of the force. Whereas the high frequency features change with movement onset, and then at force onset at time 0, they also change again. And so we looked at this in a different way in collaboration with Chetan Pandaranath's group at Emory. Chetan came from the Shenoy lab and developed something called LFADS, if you've heard of that, local latent factor analysis with dynamical systems. So modeling, in their case, spiking activity as dynamical systems using what's called an autoencoder. So they train 2 recurrent neural nets simultaneously, going from the neural activity to latent factors, and then, which is the encoder, and then going from the latent factors to your output, in this case, kinematics or force. And so here we showed with Chetan that this can also work for field potentials, for ECOG in this case. And what's nice about this technique is that it cleans the neural data. It's not really clear how that happens. He couldn't explain that to me, but it cleans it. So the insets here, these are 4 different data sets from different subjects, and the insets here are just doing straight up principal components analysis, PCA, if you're familiar with that. And you can see that we've colored each one of these lines here, which is a single trial, according to the state of the trial. So each one is a trial evolving in time. Red is pre-movement, cyan is movement, and blue is the force stage. And you can't really separate them very well, except maybe in this case. But if you use the LFADS and then do PCA, you get a nice separation. And you can see that the movement phase is clearly different in neural space than the force phase, suggesting that they are encoded differently, which is interesting neuroscientifically because it's using the same finger, but also interesting from an engineering point of view in that if you're trying to come up with a grasp BMI, for example, you need to know not just how much do I need to close my fingers to pick up this glass, I have to know how hard do I squeeze, and you don't want to, and so force control is really important. So you could think about developing hybrid decoders using this, and in fact, we got 90% accuracy in decoding state, which is not amazing, but it's it's just a first try at this, and so you could easily develop a hybrid decoder that way. So far I've talked about BMIs for replacing function, and that's helpful for people mostly with tetraplegia or locked-in syndrome, or perhaps prosthesis, amputees. But fortunately, those, except for amputees, are small populations. If you're talking about it from a company point of view, that's not so fortunate because they like to have big markets, right, for these things. So if you could use a BMI for rehabilitating function by driving plasticity in some way, this could open up the market to patients with stroke or TBI, which is a much, much bigger markets. So this is not a new idea. People have been doing it with non-invasive EEG or sometimes MEG for over a decade with varying amounts of sophistication, but there have been some higher quality studies that have shown potential for improvement when you use the BMI to control some sort of haptic feedback to the hand. That's either using a haptic device such as this one from the Ocon's group, or using functional electrical stimulation of the impaired muscles in the arm. So again, this is for stroke. So there's some promise there, but it's not clear how that improves function. It's just most groups have just said, well, we're going to have them practice imagining moving again, and somehow that'll magically improve what they're doing. We wanted to try to be a little more principled. So if you go back to some work from Randy Nudo in either rats or monkeys, he looked at stroke recovery and what happens in the cortex when that happens. So he created what are called motor maps by stimulating at all these little white dots, and then seeing what part of the body, what part of the forearm this moved. So hopefully, maybe you can see back there, but there's a maroon area, that's the digits, there's the wrist in green, and then there's proximal areas here, and then gave them a stroke-like lesion in the cortex, forced them to use that hand a lot, and the function improved in these animals. And when the function improved, they also saw that the area related to that improvement So the digits or the wrist expanded. And so this motor map expansion, while not perfectly consistent all the time in every study, was a general theme. It's not clear entirely whether it's cause or effect, but it does give a plausible way that perhaps BMIs could be used to improve function. So for example, if you consider this in a human, if you consider this a motor map, except in this case I'm defining it using recordings instead of stimulation. So this is colored by the correlation with force. This is just a schematic, it's not actual data. And if you had some subcortical stroke that blocks the downward signals from that area of the cortex, and then somehow with BMI training expand that map to other areas that still have remaining connections downstream, perhaps you could upregulate those connections. So how might this work plasticity-wise? Well, most plasticity that we know about is Hebbian, right? So, so that requires pre- and postsynaptic synchrony of activation. And so the BMI is recording motor signals from motor and premotor cortices that reflect the motor intent, and then give sensory feedback to either via the thalamus directly or primary sensory cortex. And so you could potentiate perhaps these synapses if you're giving haptic feedback. But the idea is you're only going to get plasticity if you get simultaneous activation. And so my theory is if you want good synchrony, you need good temporal precision as well. And again, what most people have been using are these EEG signals, which have limited bandwidth. So about 30 hertz is the bandwidth of EEG. And that's these blue signals, that's a power decrease down here. And what this is showing is a spectrogram just of a flexion in black, a flexion and extension movement of the fingers. And if you look at the high gamma signals, on the other hand, they're highly phasic and much more temporally precise. And so if you've got a more temporally precise signal, then maybe that can drive better plasticity. So maybe you want to be invasive and record from the surface of the brain. But before we do that in stroke survivors, we wanted to get some evidence that this might be feasible. So we took advantage of traumatic brain injury cases where they needed to have a hemicraniectomy. So they had so much swelling that they had to take off part of the skull and leave that off for months while it swells out, because otherwise they would herniate and die. So this procedure is done more commonly than you would think, but not that commonly. And this was first pioneered by Brad Wojtek and Bob Knight, who showed that you can get some high gamma signal from that. And so Mukta Vaidya, postdoc in the lab, showed that we can actually decode the thumb flexion force with the high gamma activity recorded at the scalp with EMG artifact removed with pretty good accuracy, approaching, in some cases, what we could do with epidural signals. So variance accounted for of almost 0.4 in some cases. Not always the best. And so we were really eager to use this as a platform for BMI with using high gamma to see if we can drive plasticity in the brain. And so I'm going to show you a video here of this participant here controlling, that's hard to see, but this is a picture of a thumb, and the top part of the thumbnail is the color that matches the target force. And then she's trying to match that with the color on the bottom half of the thumb. And we did color because we didn't want them looking up and down to give maybe some eye artifact. And so she's also using what's a hybrid BMI, where she's controlling the force partly with the BMI and partly with her actual thumb movement. And we've got this hybrid because we want to be able to titrate as they start getting better to have them use more and more their actual thumb, because we think that will help improve more. And so this is a short video of her controlling it. And it's hard to see, but there's a yellow outline that occurs when she improves. Unfortunately, I didn't have as much video of the good trials, naturally, as I would like. But trust me when I say they did actually learn how to use this. We've got three TBI subjects that did this so far. And then we had some control subjects as well with intact skulls that did this with low frequency signals. Unfortunately, this population is just very difficult to work with because their cognitive improvement often lags behind their motor improvement. And so we weren't able to get multiple days of BMI with as many subjects as we wanted to. But we did see, though, that clearly that the temporal precision is higher. So this is showing that here. So the black trace is the force trace. These are average signals. And the red is the high gamma power. So you see that there's a very short lag between the peak and the high gamma power and the peak force versus the mu power, which the nadir, which is the movement-related information, is much greater lag. And so you can get much better temporal synchrony during BMI use with a high gamma signal. So this supports our hypothesis that maybe it'll drive plasticity better. And then we have some tantalizing results from some of our the subjects that did finish. This is the motor map in the hemicraniectomy here, colored again by correlation with force. And so this is the BMI subject. This is a control patient with TBI that did not do BMI control but got standard rehab. And you see that the motor map did expand in this BMI subject, but not in the control. And in fact, if we look at a histogram of the change in the correlation across all these electrodes in our 2 BMI subjects, it was high. It is generally trending higher than control. So there's suggestive that that this may actually work. Okay, how much time do I have? So so just to touch briefly on our work on speech BMIs, we've done some work with trying to decode speech directly from the brain to help with communication for people that are locked in. Again, this is also not news, but this is why BMIs were initially worked on in people. But they've always been really slow. This is a diagram from Emily Mugler of relative communication rates from speech all the way down to the highest reported BCI speed until 2021, which was about 10 words per minute. And then last year, Frank Willett got a lot of deserved press for trying to decode attempted handwriting, but even that was still only about 20 words per minute, which is still slower than an eye tracker. And still much slower than texting or handwriting, which are in turn much slower than speaking, which is what I always tell my kids, just call me, it's a lot faster. But so if you could decode speech, intended speech directly, you still could do a lot more natural communication. People have been trying to do this for a while, usually at the phoneme level, which is the smallest speech sounds, because there's a finite number of those. And so most early attempts were trying to decode phonemes from motor cortex or thereabouts. But in linguistics, phonemes are not that simple. Phonemes are actually invariant representations and are based on transcripts. So they're based on perception. And so a P there is the same in any context, but that's not true of production. And I'm going to try to play this for you. If you hear this, this is going to be 3 words. Hopefully you can hear it. Oop.
Bat, spat, bat.
Okay, so who thinks the third word was bat? So the third word was spat with the S taken off. So it was really a P, but it sounded like a B. It was produced like a B, and you perceived it like a B. But if I said spat, you would perceive it as a P. So it's much more complicated than just, well, we'll just decode all these sounds. And so in collaboration with our linguistic colleague, Matt Goldrick, we looked at what some would say are more fundamental units of speech, which are articulatory gestures, so which I think of as poor man's kinematics. So the opening or closing of the tongue, tip, body, lips, palate, and larynx. And so we made the analogy that if you're looking in motor areas, you'd probably want to decode motor movements, rather than sounds. And we made the analogy to limb movements, where M1 preferentially encodes low-level things such as kinematics, force, or arm muscles, depending on which religion you belong to, and premotor areas such as premotor or posterior parietal areas encode the targets of the reach. And so we made the analogy that phonemes are like acoustic targets, gestures are the movements you use to get there. And so we hypothesized that there should be a gradient of encoding patterns in these different areas, M1, premotor, and inferior frontal gyrus, which is part of Broca's area. And so Emily Mugler, a postdoc in my lab who's now at Facebook, recorded M1, premotor, and inferior frontal gyrus ECOG from 9 different subjects, and then took the electrodes in each area and decoded either gestures in red or phonemes in blue, and found that indeed in motor areas as well as some of premotor cortex, gesture decoding was better, whereas as you move anteriorly to inferior frontal gyrus, you don't see that difference. We looked at this in a different way using what are called allophone sets of words, so words that start with either P, B, or SP, and then built decoders based on the words of Ps or Bs, and then saw how they decoded SP words, with the idea being that if an area was more gestural, if an area was more phonemic in quality, they would behave more like the Ps and decode better on the P decoders. If it was more gestural in quality, it would decode more like the Bs. And indeed, we found that from posterior to anterior, they started out more gestural and became more phonemic, moved more towards the blue, and less gestural, away from the orange here. So again, there's a hierarchy, and speech motor areas are homologously organized to limb motor areas. I probably should leave some time for questions. I'm going to briefly mention that we are looking right now at using ECOG to inform us about and understand better and perhaps improve upon functional brain mapping, which uses direct electrocortical stimulation, as you can see here, to identify so-called eloquent areas of the cortex, meaning areas that control movement or speech or language. But even though this has been done since Penfield's time 80 plus years ago in basically the same way, it's really not clear what's going on there. We don't really understand. You're zapping it with totally non-physiologic amounts of current, and it's not well understood what's happening. Moreover, it's not clear why when you zap one little spot, you can cause a speech arrest where somebody stops speaking or they make a language error, like they say ball when they mean bottle, but you stimulate 5 millimeters away, nothing happens. Whereas if you look at the ECOG or the fMRI when somebody's talking, that whole network lights up. So what's so special about those areas? We think it's something to do with the connectivity of those areas, that the highly connected, well, not just highly connected, but specifically highly connected patterns may help us identify those and perhaps one day replace stimulation or enhance it. And so we're using network neuroscience. I don't have time to go into the idea of graph theory, but the idea is you've got nodes, which are electrodes, and you've got edges, which are the functional connectivity between them. You can partition them into communities, which are more similar to each other in their activity, and then you can come up with different metrics to describe them. But I'll jump to the conclusion, which is that these critical nodes in speech, and especially in language areas, have high what's called participation coefficients, so they're connectors between communities, but they are actually low connectivity locally and globally. So there's a very interesting dynamic there, and we can actually, so Jason Shea is a neurosurgery resident in the lab, and he's been actually showing that you can actually get pretty good decoding of these critical nodes just using these basic features. With pretty high sensitivity with, again, this is very much a first pass at this. So to summarize then, talked about our work on MINT conditioning for stroke rehab, our BMI work to replace motor and speech function, talking about grasp and force, encoding speech and limb motor areas homologously organized, BMIs for rehab, and a little bit about functional mapping. So I'd like to thank my lab members and collaborators in funding. Questions?
Great talk, Marc. Really fascinating. So back to that study where you decoupled and the spiking on the same electrode. You know I was going to ask about that.
Yep.
So they're, just so I understand, they're highly correlated when they make a hand movement. That's what the one graph is showing, and they could be coupled in when they were in the BCI test. So what do you
Those were on those particular electrodes, yes. So there were a lot of other electrodes where they weren't so highly correlated to begin with. We chose to control, we initially chose ones that were highly correlated with hand movement so that we thought they could modulate them better. But yeah, but sorry.
What do you think? What strategy do you think they're using to?
I don't know. That's what we're trying to figure out. And we're trying to use some ensemble, dimensionality reduction techniques to try and see what's happening in the manifold. They're just trying? I think they might be just trying to separate one neuron from the manifold somehow. But that's what I'm trying. That's my side project that I'm doing since the student graduated years ago.
So it's not that you always see correlated gammas.
Right. Right, so even that is interesting, I think, but yeah, so thanks.
Other questions?
Yes?
I had a question on your study with the myoelectric computer interfaces. You mentioned that the preliminary results look like there might be some improvement in group follow-up, where we don't usually see that with other interventions. And I was curious what you think is unique about this therapy that's leading to that improvement in follow-up.
That they continue to improve. I think they're probably just using the arm more. It's now that they've loosened it up a little bit, so to speak, I think they're just using it more in everyday activities, and so perhaps, just more training with it. But I can't say that for certain, but that's my speculation. And I don't know that I would say no other therapies show that other therapies can, but it's encouraging that it does happen. So on the other hand, even if it didn't, the nice thing about it is that it's a home-based therapy, so you could easily think about having a tune-up session every week or two, just to maintain it if you needed to. So.
I actually had a question related to that. So I noticed that your improvement curves in the myoelectric interfaces had these interesting jumps. So because you grouped it by sort of the first 2 weeks, the middle, and last 2 weeks.
And they're different muscle pairs.
Oh, they're different muscles.
Yeah.
Okay.
That's why we separated them.
Okay,
Yeah.
got it. Yeah, because I was interested that there was both a global learning curve, but then there also seemed to be individual learning curves within weeks.
Yeah, well, so yes, and it's hard to know about global learning because different muscles, and each patient was customized to their own sets of muscles. So it's hard to know whether there's really savings there or not, to use the motor learning terminology. And plus, what this doesn't say is that it's actually a weighted time to target because difficulty levels constantly also leveling up as they get better. So that complicates it too. And are we weighting it correctly? We just assumed a linear weight, but that may or may not be correct. So there's a little nuance there. So yeah, but happy to have feedback about the best way to show that because it's not entirely clear. Yeah.
Pretty related to this, but it looks like in your multi-site trial, or at least in your multi-group trial, you had a 3D control, then a 2D and 2D plus movement. It seemed like 3D was the best, and then 2D plus movement seemed the worst.
Yeah,
that's what I'm following about. Charles asking, is there any intuition about those results?
Yeah, and it's still early because there's only about 8 in each group so far, and we're aiming to have about 14 or 15. But yeah, I'm not sure. Well, so the 3D I think it's because so we choose the muscles, and I had to gloss over this. We choose them, the first set, as the most abnormally coactivating muscles, so the highest correlations. And the 3D one gets 3 weeks with that set because initially when we designed the trial, I figured, well, they're doing 6 total muscles, and the other ones are also going to do 6 total muscles, but I think they're getting more time with it. And interestingly, in the 3D, not all of them actually get to the third muscle, or they get to it, but they don't do very well with it, some of them. And so I think it's just more time with those muscles, really. Why the reached group does worse, I don't know yet. I was surprised by that and perplexed. I don't know if it's because they're doing more loading of the shoulder and that causes more coactivation, so it's just harder to do at that point. But I don't know. That's a great question.
The reach is sort of out of plane of folks to extend their muscles?
Yeah, well, I mean, we encourage them to move in the normal groups anyways, but as you saw in that video, they often kind of make very small movements. So we're encouraging them to reach as much as they can, or at least close to as much as they can without getting too tired, which is also a big problem. But so maybe it's fatigue. It's something we have to look at. Yeah.
Last question. Yeah, go ahead.
I wanted to ask something related to that. I wanted to ask, how do you think about scaling sort of the dimensionality of these tasks? And how do you think, with the 3D performance, like the performance on higher dimensional tasks might be related to the variance available to the participant to begin with?
Yeah, that's a good question. So when you, let me understand, when you say variance available, meaning all they can do is this?
Yeah, it's the component breakdown that you showed us, and how quickly you can capture quite a lot of that variance.
Well, so we are looking at the so-called muscle synergy analysis of that, of what's happening in that, and we have a paper in review that's looking at our older data with that, and suggesting that they don't actually change the whole so-called synergy, or, you know, it's not really PC, but it's an NMF, but they don't. It's pretty limited to the muscles that are being trained. So which is kind of interesting that the brain can, or the CNS, we don't know where it's happening, can change on such a fractionated level even after stroke. And so yeah, so we're very interested in figuring out where is the plasticity occurring. But in terms of 3D, yeah, I don't know. I haven't thought about it in that way, but I think it's just whether maybe it's not, maybe it has some fractionation ability, but not, but not that much. So I don't know, but it's a good question. Yeah.
All right. Well, let's thank Dr.
Thanks.
Slutzky.